Chapter 4, Cortical Organization. So, there is an electronic map of the retina on the striate cortex, which is the visual cortex located in the back of our head, right about here, right? And this map is referred to as a retinotopic map. Okay, so retino, of course, refers to the retina. And what this is saying is that for any particular point on the retina, so again, imagine the retina as this round sheet, right? For any particular point on the retina, there's going to be a corresponding point that is connected to that receptive field on the retina in the cortex, on the visual cortex, right? So there's this relationship, there's this mapping of the space of the retina mapped onto our visual cortex, and this mapping is called a retinotopic map. Um, there's also this concept of cortical magnification, meaning that there's more area of our visual cortex that's devoted to the fovea, which is the part of the retina where we uh, where we focus on details, right? So whenever our eyes are uh, uh, focused on something, the center of our focus is always projected onto the fovea because the fovea contains the most cone receptors, so that you get the highest density of cone receptors there, so you get the greatest resolution, the greatest attention to detail in the fovea. That's also true in the cortex. There's more area of cortex that's devoted to processing the information in the fovea than there is in the outer part of the retina, and we're going to see this for other senses as well. For example, for touch, we're going to see that there's more area devoted to the hands and to the lips, which are very sensitive to touch, than, say, the elbow, which is not very sensitive to touch at all. There's more of those touch areas in the cortex for the lips and for the fingers than there are for other parts of the body that are less sensitive to touch. So the example from your book here is kind of an illustration of the retinotopic map. We see this person with the unusually gigantic eyeball looking at a tree with 4 points defined on the tree, A, B, C, and D, and those points, of course, are projected onto the retina and light up on the retina as A, B, C, D. And corresponding to the points on the retina, those same points are represented in the cortex as cortex points A, B, C, and D here. Um, this example here, this photo here, gives us an idea of the idea of cortical magnification. So in other words, the fovea, which is right here, this little part of the retina right here, only accounts for less than 1% of the area of the retina. So the fovea is just this little spot here, and then we have the entire retina here. So that's not too much of the space of the retina, but look how much of the visual cortex is devoted to the fovea. 8 to 10% of the cortical map of the cortex is devoted to fovea. And of course, the reason for that is because there's so much information coming from this part of the retina that there's more cortex required to process that information. Um, One of the ways that we know how visual processing works in humans, in live humans, is through the use of different brain imaging techniques. So one of the most popular brain imaging techniques is PET scanning, positron emission tomography scanning. And this is where we can actually see the activity of the brain uh, while a person is doing a task. So we can have somebody looking at something or thinking about something or listening to something and record the movement of radioactive ligands in their blood through their brain. So basically, the way it works is you get injected with a radioactive tracer. Now this radioactive tracer is basically sugar, glucose, with a small radioactive um, um, molecule stuck to it. Right, so it's a very low radioactive molecule. It's not highly radioactive, but it's just enough to be detected with a very sensitive instrument. So the tracer gets injected, it goes through your bloodstream, and then it goes to the brain, of course. Now the brain is the most richly uh, uh, um, blood supplied organ in the body. So as the radioactive tracer makes its way into the brain, the parts of the brain that are working the most consume the most sugar that has the radioactive tracer. So we can see differences in the areas of the brain that are working harder than others simply by tracing how much radioactivity gets consumed inside those different parts of the brain. And so we can see changes in the way the blood is flowing, and we correlate those changes in blood flow to changes in brain activity. Um, and the way it works is first you measure the activity when they're not doing something. So when they're not looking or focusing on something, you measure the activity. And then you have them do whatever it is you want them to do or look at. And then you measure the activity while they're doing it. And then you take those two measurements, those two radioactive measurements from each part of the brain, and you subtract it. And whatever differences there are, we presume that that's because those parts of the brain are working during the time that the brain is being stimulated. And this is called the subtraction method for obvious reasons. Another method that we use besides PET scanning is fMRIs, functional magnetic resonance imaging. Um, Basically, what we can do here, um, we can trace the flow of the oxygen and hemoglobin inside of the brain. So hemoglobin, which is a large molecule in our blood that is responsible for carrying oxygen to all our cells, actually contains a bit of iron. If you've ever been anemic, it's because you don't have enough iron in your blood, which means that there's not enough iron to go around in your body for the hemoglobin to there's not enough iron to be incorporated into hemoglobin to carry enough oxygen, so you end up being kind of weak and anemic. That's what causes that. But hemoglobin has a little bit of iron, a little iron molecule that is, of course, magnetic. All iron molecules are magnetic, right? And of course, the hemoglobin with this oxygen and iron molecule makes its way to the brain and it gets sucked up. Um, Once the oxygen is removed from the hemoglobin molecule, the hemoglobin becomes more magnetic. So the change in the shape of the molecule makes it more magnetic. So we can actually use this difference in uh, oxygen carrying and non-oxygen carrying hemoglobin molecules to see which brain areas are most active, right? So kind of like we use the same subtraction technique that we use with the PET, right? So we put somebody into an MRI machine and have them, think of something or look at something, whatever parts of the brain we're interested in studying, and we measure the activity of their brain. In this case, we measure the hemoglobin being taken up, or the oxygen being taken up by different parts of the brain. And then we stimulate them, right? Have them look at something or think about something or whatever. And then we subtract the difference. We take the measurements that we got in the machine from when they were being not stimulated and stimulated, subtract them, and use that to determine which parts of the brain are more active while they're looking at something or thinking or whatever, right? And here's a picture of somebody inside of an MRI machine, right? Lying very still, of course. So one of the things that we've been able to determine here, and if you look at this picture, we can basically what we did here was we put somebody in an MRI machine, and if we look at this part of the cortex here, so this is the this is a this part of the this picture here is Um, if we took a human brain and sliced it down the middle, and then we're looking at the inside of the brain here. So if I were to take my brain and then slice it down the middle like that, and then take this half and show you the inside, that's what you're looking at here. So this is the back of the brain, the front of the brain, the top of the brain, right? And this is the inside surface here. And this is the back where the visual cortex is located. And what we did was we put somebody in an MRI machine like here, and then we had them look at this stimulus here. We had them look at this little red stimulus and this blue stimulus, which are arcs on a circle. And using MRI, we were able to detect the parts of the brain that were activated while they were looking at each one of these stimuli independently of each other. So like I said, the cortex is organized into columns, right? So each spot on the cortex handles a certain receptive field, a particular receptive field that corresponds to a spot on the retina, like we mentioned earlier, right? However, if you go down the cortex, go down into the cortex in this particular spot, you'll see that the cortex is organized into columns. The visual processing system is organized into columns in the visual cortex. And of course, a lot of the visual cortex is devoted to the fovea, right? Studying or processing the information that comes from the fovea, and this is the cortical magnification factor. Here's an example, right? So this right here, again, is an example of the visual field of what we're looking at, right? And let's say we're looking at this, and this little dot right here is what we're focused on, right? If we were to then look again at one of these half chunks of the brain, right? So once again, we have this half chunk of the brain where, again, I took the brain, cut it in half, and showed you the inside of it, right? So this is the back of the brain, your forehead is here, this is the back of your head, the top of your head, right? And this is the inside of the brain. Once again, we're going to focus here on the visual cortex. And if we look at the way the columns are organized, the receptive fields are organized in the visual cortex, we'll see that this little dot here corresponds, if you're looking at this visual field, this little dot here would, and if you're staring right at this little dot, this would represent the information that's going to your fovea if you stare right at that dot, and all this stuff would be going to the outside part of the retina. And if you look over here, the way it's mapped on the cortex is very interesting because you see this huge dot here. So we have this whole bunch of processing, this whole bunch of space right here devoted to this little tiny area right here. And then as we move out into the visual field, to the edges of the visual field, you'll see that there's less and less space devoted in the striate cortex, the V1 striate cortex. If you look at the visual cortex, and this work was originally discovered by Hubel und Wiesel, right? So these are the same guys that we talked about in the previous chapter, in chapter 3. This was their original work that they did back in the early 60s, and one of the things that they identified in their original work were these columns, right? The visual cortex has these different columns, and they are literally shaped like columns, like tubes, okay? So you can think of these as tubes so if this is the surface of your cortex, you can literally think of them as tubes that are oriented perpendicular to the surface of your cortex, right? So you have all these tubes or columns that do different things, right? So for example, Hubel and Wiesel found location columns. So if there's a particular receptive field on the cortex, the column underneath that receptive field would light up with different locations, different orientations in the same location, right? So everything was focused on that particular location. There were orientation columns, so inside the cortex are orientation columns. So inside of each orientation column, it's made up of neurons that fire with the same orientation of stimuli. So there were columns that would fire when the stimulus was oriented vertically, when it was oriented at different angles, right? And all the columns are lying next to each other. So one column fires when a bar is oriented vertically, then the column next to it fires only when a stimulus is angled a little bit more, the one next to it a little bit more, the one next to it a little bit more. They're all lined up very sequentially next to each other. Um, and they could move an electrode about at 1 millimeter, which is a tiny little amount, about that big. And if they move the electrode around a 1 millimeter, they could record the entire orientation of a particular area, right? So they would move the electrode that they're recording from. It would go from 0 to 1 millimeter, and as it moved across that 1 millimeter, they could record different cells that would respond only when bars were oriented in certain positions, which is really interesting.